All right, let's go. Hello, we're doing second sequencing today. Now, we did DNA profiling and PCR last lesson. So we learned that PCR is just the process we use to get lots of copies of a single piece of DNA, right? And profiling is when we start to separate that DNA into bands, like on an agar-agar gel for gel electrophoresis. DNA sequencing is a bit different because in profiling, we just saw the bands, right? So we actually don't know the sequence of DNA, right? AT, GC, whatever. But in sequencing, you're trying to determine the sequence of nucleotides, right? So what exact order they appear in, right? So what I'm going to do today is I'm going to explain the process of second sequencing and show you how it works. The TLDR is, yes, the target DNA is copied many times and fragments of different lengths are made. And then we have these special nucleotides that play a different role, which help us determine the sequence. We still use gel electrophoresis, and we still use like some kind of PCR, but it's got a few different steps, which is why it's called a different process. And those steps help us to figure out like what exactly is going on. So, how is it similar, right? Well, we still use an enzyme, right, polymerase, to make more copies of DNA. We still have a primer because the polymerase needs to read the primer to start laying down nucleotides, right? We still need template DNA, but I guess the difference here is we have these special things, right? These 4 special DNA nucleotides. They have these weird names, right? And what they are is these things, right? So you don't need to know the chemical structure, but functionally they're the same. So like a T version of this, right? DDTP will still pair with an A, just like a normal nucleotide would. But the difference here is that they're chain terminating, which means that after one of these 4 nucleotides pair up, the chain can't keep going, right? It just stops at that particular length, right? So I'll find a way to show you what I mean, right? So let's say I have a little vial, right? And then inside my vial, I'm chucking in what's going inside? My primer, my polymerase, right? My normal nucleotides. I'm going to bracket normal, right? Nucleotides. Right, so this is still PCR. Everything is exactly the same, right? But now I'm also adding I'm going to call them special nucleotides. I don't want to write the full name. Chain terminating nucleotides. Right, cool. So I have all this in my solution. Right now, I start PCR, right? So we know PCR, we have like denaturing. So we've got to heat the solution up to break apart the hydrogen bonds. And then we cool it back down so that we can apply the primer. And then the polymerase can start laying down nucleotides. So all that's the same so far, right? So I'll just draw what that looks like on the board, right? So I have my DNA chain. I've broken it apart. I've denatured it. I've gotten this, right? My primer has started. I'll denote that in green, right? Okay, cool. So the difference here is, right, when my dideoxynucleotide binds to the DNA strand, it will terminate the chain, right? And they're also attached to a fluorescent dye. So I'll explain what that means, right? So the video, the image explains it pretty well, but like I'm extending my chain, right? I'm adding normal nucleotides, but then suddenly, right, instead of adding like a normal nucleotide, I'm adding a special chain terminating nucleotide, right? So let's say I add one that's this color, right? after I add one of these nucleotides, this chain, it stops growing. It doesn't get any bigger, right? That's it, the chain, right? Now, the same thing can happen for another piece of DNA, right? So normal nucleotides are being added, right? But then again, we hit another special chain terminating nucleotide, and then we stop again, like this, right? So then what you'll get is, obviously, because you've stopped early, you haven't made the full length of this DNA strand, right? Because the chain terminating nucleotide means that like you just didn't get to the end, right? Because nothing can pair after this thing attaches. So what you get is, well, you have to repeat the process because you have to keep trying, right? So what you get is you just get a bunch of, I'll just do 5 examples, right? So let's say my first example, I get like 2, 3 length, 1, 2, 3, 4, 1, 2, 3, 4, 5, 1, 2, 4, 5, 6. All right. Yeah, do this thing. All right, cool. So let's just pretend that like on this one strand, I hit the chain terminating nucleotide on the 4th base, right? And on the 2nd strand, I hit it on the 5th base, and then this one on the 6th base, and then this one on the 7th base. 1, 2, 3, 4, 5, 6. Oh, sorry. Yeah, this one's on the 7th base, right? Right. So, and so on and so forth, right? Can we see how we get this? Yeah. But then you also might notice, okay, we can actually use this to find out the order because, as you might have figured out, all these strands are different lengths, and we know we can separate things out by strand length with the use of gel electrophoresis, right? So what we do is once we've run our PCR and we've gotten all these different chain lengths of DNA with a special nucleotide that glows at the end, we then chuck it into some fancy DNA sequence, right? And then we get something like this. Yes, Lucas, what's up?
Are the 5 examples different like parts of the DNA, or does the chain terminating thing just add on like after a random number?
Yeah, it just randomly adds on. Yeah. But I've just ordered it in terms of size, that's all, right? So what happens here, well, we chuck it in, right? So everything in this first column, the first column here, ends with an A nucleotide. Everything here ends with a T, everything here ends with a G, and everything here ends with a C, right? We go from shortest, right, because we know that the shortest DNA strand moves the furthest, to the longest, right, which is this, right? So that means my 1 would probably be maybe something like this, right? And my 10 could be, maybe my 5 is this and 10 is this, right? So even longer, right? And by doing that, we can figure out exactly what the order is, right? So just looking at this band, let me see, right? I can look at the sequence, right? So the sequence from left to right would be, right? It would be A, G, C, T, T, A, G, C, C. Right? Does that make sense? Can we see how you got that? Right? I literally just read it in order from like bottom to top.